Who is our last universal common ancestor? Who is LUCA? And that is the objective of this episode, where we are going to attempt to reconstruct and paint a picture of who LUCA was, their physiology, perhaps their age, and where they sit between the origin of life and today. And we're going to use evidence, scientific research, fairly recent paper in 2024 from Nature, primarily as the building block for establishing our picture. And what better place to start than with a brief history of how we even established this concept of LUCA. And I think it starts in 1866 with Ernst Haeckel. And he was a zoologist and many other things, which most people were in the 1860s. And in the 1800s, they took on a lot of titles. But Ernst, he established for the first time classifications for all of life on Earth. And it was plants, animals, and protists. And those funny protists, but even today, similarly to today, the protists just capture what we don't know. It's just we have obviously in the 60s, they didn't have the technology we do today to investigate these matters, but obviously we have plants, we have animals, and then I lumped everything else with protists. Ernst was actually an interesting man. He identified a lot of different species and named them, and after his first wife passed, he named one species of jellyfish after her, which I thought was interesting. But then 100 years progressed, and with time and effort and research, we were able to establish a more accurate description, and that was with the help and the work of Carl Woese, who worked at U of I and studied at Amherst as well as Yale. And he gave us the 3 domains of life, which you probably studied at some point, and that is archaea, bacteria, and eukarya. And this description of life relied on structures and features of the cell and specific things that bacteria had that archaea didn't, and specific things eukarya had that both bacteria and archaea did not. And this was probably the first time there was a scientific idea for LUCA, and the first time pushing forward this concept that isn't just brainstorming or what have you. And so we, in the 90s, we have this idea of LUCA, and then in the 2000s, this complicated and difficult phenomenon known as lateral gene transfer, or maybe in some places horizontal gene transfer, kind of muddied the waters, and there was a lot of debate and deliberation over how to handle it. And then in the 2010s, there was finally a consensus that we still use today, and that is that there are 2 domains of life, archaea and bacteria, and eukaryotes are just a chimera of the 2, whereby an archaea consumed or engulfed a bacteria, specifically a cyanobacteria, through endosymbiosis, and created this new cell that could be more complex and contain more genetic information. And we believe this to be true because the mitochondria contains bacterial genes, while the nucleus of a eukaryote contains archaea genes. And then so this idea of genes and who has what genes and where are they, we're able to go back in the lineages and identify exactly which genes are present in all of them. And this helps us establish who LUCA is. And to date, and based on this 2024 paper that was published in September, I believe, or July, we have a model that gave us 2,657 genes that are present in all archaea and all bacteria, and thus would have to be that node, that last node before bacteria and archaea branch off. And it contains roughly 2.57 megabases, or 2,570,000 bases. And for comparison, a modern day prokaryote has roughly around, on average, 4 million bases. And a modern day eukaryote has in the realm of billions of bases. It's just for a comparison there. So kind of consistent of what you would expect for something, for all cells to originate from. And so from this, we can construct the physiology, the features, the metabolism of LUCA. And we'll attempt to paint a picture of what this may have looked like based on the genes that we've already identified and that researchers have identified. And then lastly, we can look at recovered microfossils in South Africa, Australia, and control for the moon impact theory. And we can kind of pinpoint an age range for LUCA. And so that's what we'll do in the rest of this video. And so what genes are present in LUCA? To understand this, we have to understand the methodology the researchers used. And what they did is they took samples from the KEGG orthology database. And this system just is like a mapping of all the genes that have been studied already, and they have the assignment of which organisms they are present in, and then you can see which genes are absent in other organisms, and then you can look down the lineage. And they take these samples, the researchers took these samples and funneled them into their algorithm that reconciled out which genes would be present in LUCA and which ones wouldn't be, based on a probability, so it's not certain. And the probabilistic nature of this algorithm means it can only be so reliable. I don't know how reliable it is, but I'm using it to construct our picture, and with time the algorithm will probably get better. With more computing power and more effort, it'll likely get better. But for now, it is what we'll use to establish our picture. And so what is that picture? That picture is a life form that is roughly 4.2 to 3.8 billion years old. And to establish this age range, we have microfossils that give us an age estimate for how young it could be, and that is roughly 3.5 billion years ago. We found microfossils in Australia, although there's controversy over whether it's truly a cell, but it does have complex organic molecules. And then there was microfossils, almost assuredly a cell with complex organic molecules, established 3.2 billion years ago in South Africa. And so we can use that as our lower estimate, and then we can raise it up to 3.5 billion years of age. So that's the youngest we think LUCA could be. The oldest it could be is roughly 4.2 billion years, based on the moon impact theory, where two very large objects collided with one another roughly 4.5 billion years ago, and the product was the moon and the Earth. And so life couldn't have sustained itself until maybe roughly 200, 300 million years after that collision. And so that's how we get 4.2 billion years of age. And now that's much older than I thought LUCA would be before I started investigating this topic. And so, as you might imagine, the atmosphere, the climate, the entire composition of the Earth was much different than it is now. And so that is the guiding principles or factors that went into my creation of a 3D model of LUCA, which we can look at now. LUCA, our last universal common ancestor, dwelled during a time when the atmospheric composition was primarily carbon dioxide, methane, nitrogen, and most importantly hydrogen gas, which came from volcanoes and metamorphism. And it gives a red tint to the sky. The temperature is very hot, it's 49 to 88 degrees Celsius. We are at the ocean surface and not hydrothermal vents. The environment has organic compounds and molecules such as sugars that LUCA will use as energy. So let's take a closer look and dive deep into who LUCA was. LUCA has peptidoglycan surrounding the cell membrane, and it's gram-negative as we can see here because it was stained a color that is pink or red. There are actin-like protein filaments making up LUCA's cytoskeleton. There is a single circular chromosome with roughly 2.75 million base pairs, and there are ribosomes within the cytosol or cytoplasm available to conduct translation throughout the cell. LUCA's metabolic pathways such as glycolysis and carbon fixation, or more specifically the Wood-Youngdahl pathway, along with a modified TCA cycle, rely on a network of enzymes and complexes that facilitate the energy transfer. Not to mention there is an ATP synthase and a minimal form of the electron transport chain. Hydrogen gas diffuses through the cell and into nickel iron hydrogenase, where the gas is split into 2 protons and 2 electrons. Additionally, 2 molecules of carbon dioxide also diffuse through the cell membrane. One goes through a series of enzymes and complexes and is reduced with the help of electrons from the hydrogen gas that was split. The other is converted to carbon monoxide by carbon monoxide dehydrogenase. They meet together with coenzyme A to enzymatically form acetyl-CoA via acetyl-CoA synthase. Meanwhile, glycolysis is also going on and forming acetyl-CoA. Acetyl-CoA enters the 3-enzyme modified TCA cycle. It meets with a 4-carbon molecule at homocitrate synthase. Out comes a 6-carbon molecule that enters succinate dehydrogenase, which spits out a 4-carbon molecule that enters 2 subunits of ferredoxin oxidoreductase. This all leads to the advent of ATP and the generation of a proton gradient that powers our limited ATP synthase that has only beta subunits instead of beta and alpha subunits connected to the gamma subunit.